BR biosynthesis, jasmanic acid, or also ethylene, which are affected differentially. And you can see in heat maps showing that overexpression causes downregulation of these genes, and they are upregulated in case of controls. Now, once we sent the data for publication to Plant Physiology, they wanted us to show that one of the genes we claimed is downregulated is this KO2, which is involved in actually initial steps of biosynthesis of GA from this particular general pyrophosphate to GA12, and it is involved in regulating 3 steps of the hair, causing the formation of kyurinate, which finally goes to form GA12 aldehyde. Finally, you have active GA1 and GA4. And we were able to finally show in the promoter of this particular gene, there are 3 light responsive elements. Out of that, one of them is the one which is actually binding to that particular high 5 and also show reduction in the movement of the fragment during our EMSA. And when we mutated that, they were not able to cause reduction in the movement, showing very critically that this high 5 ortholog in rice is able to bind to the promoter of KO2 and regulate the biosynthesis of GA. Maybe because of that, you have reduction in the plant height. Now, another question came, as I told you in the very beginning in the introductory slide of Xinguang Dang, this particular protein is actually degraded in dark and also is stable in light. That's why it's able to regulate photomorphogenesis. Question came, what happens to BZ48 in case of rice? Is it light stable or is it stable in dark or not? Now, here is an experiment which I think very clearly shows that this is now a 4-day-old seedling grown in light or dark and then exposed to dark in this case and move on to light for various hours. We did not find any change in its overall expression profile. These 2 bands show that one of them is actually a form which is phosphorylated, which is well known in case of Arabidopsis also. So this is in contrast to Arabidopsis, because in case of Arabidopsis in dark, when you transfer plants of Arabidopsis from light to dark for various hours, you find there's a degradation of this particular protein. And that's the reason why it does not differentiate in case of darkness, but then as again from dark you should expose it to light, you're able to actually recover its accumulation as the protein. Now, the question came from examiners again, who were reviewing our paper, that you have shown that in case of rice, this is a stable protein both in light and dark. You have also expressed this protein in case of Arabidopsis. Is it stable there or not? Does it behave like Arabidopsis or is it also behaving differently? So this is that experiment which shows very clearly this 48 gene expressed in Arabidopsis also is expressing a protein which is not again degraded in Arabidopsis. Now we have shown that this degradation occurs because of the E3 ligase, which is COP1. The COP1 in rice, in fact, does not interact physically with 48, and as a result of that, this protein remains stable. So this particular whole data which I presented is now due for publication. In fact, it should have appeared about in the month of September, but the editors of Plant Physiology advised that we have a focus issue coming in February next year, and with the title Energy, Focus on Light and Oxygen, whether would you like to put this paper in that particular issue of that, and we agreed for that. So all this available online, but it will be now the whole data I presented will be appearing in Plant Physiology, published by the American Society of Plant Biologists. And so in conclusion, I would like to say that although the Arabidopsis mutant we have been able to rescue using rice gene BZ48, but when you overexpress that in case of rice, we are able to cause reduction in height, which is in contrast to what happened in case of Arabidopsis. Also in case of rice, when we knock this gene down, we have internode elongation. But then if you go to next generation, we have much more expression of this, down regulation of this. These seedlings become lethal, and as a result of that, the plant is not able to survive. Observation again, which is in contrast to Arabidopsis. And this, I think, is a high point of this particular presentation here, that the protein high 5 is unstable in dark, but this BZ48 of rice, which is orthologous to Arabidopsis gene, the protein somehow is stable and might be causing some functional roles which are quite in contrast in contrast to Arabidopsis. With that, I would like to conclude and thank you for your attention.
Okay, thank you very much. Any questions? Okay, I have a quick question.
Yeah, sure.
So I'm doing a wheat global transcriptomic analysis, and I'm thinking I'd like to add your target to my list of genes. Do you know if there's a homolog in wheat? Have you looked?
Yeah, there are. There are homologs in
it's-
wheat also.
It's across the board, because of
That's
course
right.
Arabidopsis is
Right.
monocot,
That's
but
right.
rice isn't.
Right.
Thanks.
Yep. Okay.
Any other question? Oh.
So overexpression of BZ48 leads to the retarded growth in rice. So you have any insight into it except for the metabolic pathway? Because I feel that since it's a BZ transcription factor, it is expected to have number of heterodimerizing partners, not as a homodimer. So probably what is happening is that you are overexpressing a protein, and now it's just dissociation constant. It's so much of it's there that is going to interact with its partner, and as a result is being sequenced out, and the genes are downregulated as a result. Is that a possibility?
Not exactly. It does form a homodimer. It does form a heterodimer with BZIP1, also with BZIP18, and we are testing others also. But what exactly is known is now, because when you talk about reduction in height, ultimately it boils down to control of hormone levels and hormone action. Ultimately, GA, auxins, all of those are going to be regulating that. So somewhere down the line, you have to think about that light is pursued by cryptochromes and phytochromes. They then turn on the activity of Hi5, and Hi5 eventually is able to go and bind to the promoters of all these genes. So looks like that overall plant growth and development by light is coming again at the level of hormone. And that also, also that GA also controls cell elongation as such, and some of these phenotypes I've shown also shows change in cell elongation. So I think there is a clear connection which is coming, and there is no point of any other deviation as such. So light regulates plant height by modulating these through these factors, the hormone levels, which eventually will regulate the cell size and cell elongation and controlling plant height.
I have a question. Do you have any insight into how these proteins are reacting to light or responding to light? Does it involve the aromatic amino acids? Does it involve photooxidative modifications? Once they've responded, do you think they get degraded and recycled, or is it like in bacteriorhodopsin where there's a conformational change which is reversible?
No, I think they are degraded. There is no recycling as such. But I think what light is doing, because of its prevention, there is some change in its phosphorylation status. So phosphorylation also is one critical factor.
But is that mediated by the aromatic amino acids because there's nothing else that would respond to UVB differentially? In fact, all proteins would have, there are 4 chromophores in proteins.
Right.
Unless they have something else, like hemoglobin. The peptide bond and the 3 aromatic amino acids.
Right.
So, and they're there in most proteins. So what is it that allows these proteins to respond to light differentially?
No, actually, light is being perceived by phytochromes, cryptochromes. So they are all redox changes which happens in case of other receptors also. So these are the steps which are coming in signal transduction through kinases and all that. So basically those are the things which are causing phosphorylation also. So you have to bring in, I think if I go to the very second slide I showed you,
So
there are intermediate components which will channelize the energy. So light force is working with the photoreceptors at redox level, changes are there, and that photoactivation will lead to kinase getting activated. Those kinases will work on these proteins.
Cascades of phosphorylation.
That's right. That's right.
Profiling of hormones in the contrastive behavior happened in rice and Arabidopsis.
We have not done the estimation of hormones as such. So some of those genes which we found in the microarray, we confirmed that with the real-time PCR, but we have not done the estimation on the hormones as such. Yeah.
Okay, and I think we should probably, is that working? Move on to our next, that's not working. Move on to our next, thanks, Dr. Khurana, for that lovely example of classic plant molecular biology. So our next speaker will be Dr. Banerjee. And while he comes up here, everybody stand up for a minute, because I can just see it. You're all falling asleep, and so just stand up for a minute. 60 seconds while we get the next talk ready. But don't leave the room. Well, I'll introduce you. Okay. I'll say a couple more words. Okay, and so perhaps I'll even start introductions. Thank you, Dr. Singh. That was a good talk. Train. I'll start with an introduction. So Dr. Anjan Banerjee is from the Indian Institute of Science, Education and Research in Pune. He got his PhD in biotechnology at the University of Pune with research work at the National Chemical Laboratory. He did a postdoc at Iowa State University in the US, and he returned to India as an assistant professor at Indian Institute of Science, Education and Research in 2008 and has been there ever since. And he'll be talking to us about the role of mobile RNAs in tuber development in potato.
Good afternoon, everyone. First of all, I'd like to thank the organizers, Professor Sharma, Dr. Pandey, and others for giving me an opportunity to come here and share with you the research that I do. So I'm really grateful. Unlike all my senior speakers, my lab is 9 years old. I have been working on this field of understanding RNA transport, mobile RNA. You must have by now read my title. The mobile RNAs play an activator repressor role to control tuber formations in potato. Potato is the third most important food crop, right after rice, wheat, or if you bring maize in between, then it becomes fourth. Whatever it is a very important food crop in terms of its energy, in terms of its bioenergetic cost. So we really pay a lot of attention to understand what this mechanism is and how this plant really drive the turn on the switch to make potatoes. So it's basically, Professor Khurana has described a lot of plant development, and I hope the stage is pretty set now for you to understand what is that I'm going to talk about. I'm going to talk about mobile RNAs and what are these mobile RNAs. Mobile RNA field is not very old, roughly about 10 to 12 years of old. From UC Davis, many people have done phenomenal work to show us that what mobile RNAs are. And this concept came to the field indeed in the community of plant biologists. People did not know what causes flowering. So we had an old concept of florigen from 1935. And 2005, it was first time people came back. Ove Nelson's group showed that it is the FT mRNA which moves, flowering time mRNA moves and causes a plant to flower. But this result was later disputed 2 years later in 2007, and then people challenged, and then it turned out that it is not a mobile RNA anymore, it's a protein. In last 10 years, this field has gone much ahead beyond our understanding that what are these mobile RNAs and do they plant. Why would plants select mobile RNA? And today I have glimpses of results from my lab that I would like to share with you, that in the life of potato, we clearly established that there are mobile RNAs. And the title I showed to you, the mobile RNAs play an activator repressor role, means it activates as well as it represses. So why would that do? So try to understand, and I think I'll give you some introductions in the beginning for you to look at what these mobile RNAs are. So wherever I go, people ask me a number of questions, and I like to make it is easier to see. Let's look at those questions first. What are those mobile RNAs? Why do they need to be mobile? And do they move short distance or long distance? What is this movement associated with? How is this movement, how do they move? The big question comes to the field today, how do they move, right? Do they move alone? So these answers I'm required to give you today. Otherwise you don't let me go. Does all RNA move? Question I have to answer. Are they repressed from translations or protein synthesis? Because it's a very fundamental concept, DNA transcriptions, translations. You like to see an RNA make to the protein, but not always. We have seen there are RNAs that don't really make proteins at the site of their transcriptions. So it's a very fascinating area emerging in the life of plants, and with respect to it is now basically redefining our understanding in respect to plant development, be it stress, be it defense, whatever you consider in the life of plants. Are they more stable than any other RNAs? And indeed they are. We will show you that is the case. But does small RNA move? Another level of regulator coming in, the master regulator today are the small RNAs, and we now know that yes, there are examples that small RNAs are indeed 2010 onwards, now we know that in the field of plant science that small RNAs do move in the life of in plants. So if you look at the whole plant level, this is nothing but you can imagine that this is the shoot apex and this is the root, right? These are the leaves, these are axils. So if you look at all these response, Professor Khurana has described very clearly the light and its major role in terms of plant growth and development, the photoreceptors. So now, how does this leaf perceive the signal? And once it perceives the signal, all the signals get translated to protein right up there, or there are RNAs, they go from here to to either towards the apex or towards the down? That's the question. And that is all about the story of mobile RNAs. Okay. So I hope I will be able to give you some. So this is what we say this is nothing but the super highway, because we have now seen that if you take a scan, if you take a cross sections, and if you take those cells which are called vascular bundles. Those of you know vascular bundles, it has got phloem and xylem. If you take phloem, you will find plenty of RNAs are present in phloem. But phloem is indeed anuclear. There is no nuclear machinery in the phloem. Where those RNA come to the phloem? That means these RNAs are expressed somewhere in the plant and then they go to the phloem. Now, why would this RNA molecule go to the phloem? Do they have any job associated with it? Or do they have any functions linked to it? That's the questions we are asking, right? And I take potato as a model system to understand, and hopefully I should be able to convince you that what is this concept all about, okay? This is the source and sink. This relation is nothing but, as a plant grows from juvenile to the reproductive phase, it has its flower. It's a reproductive fruit. So, fruit is a strong sink from the leaf to the strong sink. The food must go, or whatever, be its development, be its growth, be its defense. All sorts of molecules must transport from the source to the sink. That's what we say. So I will use this word source and sink. You will say that anything I talk about in source, that means this RNA must be expressed in the source, and this RNA must have transported through the phloem stream to the sink. And in potato, it is potato itself the string. So all these RNA molecules, they move, unlike animal cell, they have gap junctions. In plants, we have plasmodesmata. And plasmodesmata is the connecting gate between cell to cell. So they move from one cell to another cell through this plasmodesmata. Again, this plasmodesmata has the size exclusion limit. Not all RNA can move. Not all RNA protein complex can move. We have now evidence of all this. Okay, so if you consider the plant, you can look at how it is. It is the plant vascular system functions as the conduit for long distance communication between distantly located organs. Organs located here from root to shoot, from shoot to root communications, the discussions, the talk that plant execute without our knowledge. Okay, and that is what it is. So schematic illustrations of the xylem and phloem, illustrating the fundamental feature that in contrast to the animal kingdom, the flow pathway is non-circulatory in nature. So we are talking about this complete pathway inside the plant. This is a review. I give you a cartoon that how these RNA molecules are likely to move from cell to cell. I will again bring out the evidence later, but for you to understand, RNA is a long distance macromolecule, and this is cell autonomous RNA. I say CAR versus NCAR because RNA molecules are transcribed in a particular cell and it goes somewhere else. So that's a non-CAR, NCAR. That means it is non-cell autonomous transport, and there is a cell autonomous transport as well. This is the model organism that I use to understand this concept. If you look at this model organism, you see these potatoes are grown below ground. This potato, by the way, it's a modified stem. Now the first question comes to my mind, it's a modified stem. Why would plant grow a modified stem below the ground? Why doesn't it grow above the ground? Right? So I have an answer. If you can answer, that will be nice. So this is a modified stem which grows. You can look at this stem, how this stem grows below the ground, and it swells up. And this swells up, this organ ultimately develop to a potato. Completely reloading and loading and reloading of sucrose from the phloem after photosynthesis, it just stores in this organ and makes the big organ for us to understand. Okay, if you look this slide cartoon, it will be much easier for you to understand even better. You can look at this. This is this organ. Look at this transition of this organ. Okay, from this organ it develops it's a hook-like structure. From this hook-like structure, it gradually swells up, right? And this swelling basically allow sucrose to accumulate, photosynthate to accumulate. Lot of cell division changes, lot of cell fate changes, lot of developmental changes happens in this organ, and finally this makes a big tuber for us. Okay. This phenomenon is known as tuberization. We tell them it's a tuberization, and this is governed by number of factors. Okay, so who are these factors? I made a cartoon slides for students to understand. So the players like nitrogen plays a critical role. Photoperiod, the light duration, short day versus long day. Photoreceptors, of course, phytochrome. Temperature also plays a major role in these transitions of this organ from here to here. Endogenous factors, you can talk about hormones, you can also talk about small RNAs. We now know small RNAs also play role in this organ development. Okay, we have done a lot of work in this area as well. What are other factors? The molecular factors or the genetic factors, if you look at, the 5B, phytochrome B has been shown to play critical role in the development of potato. We also see constants, we also see CDF1, cyclic DF1, it's a protein, followed by ST-BEL5, the protein that I have worked on extensively in my postdoc, followed also, and the NOX, the master regulator of plant development, and you have SP6A, recently identified another co-regulator cofactor we have identified, another group have identified, that's also a mobile signal for potato. So in addition, you have other factors, you have small RNAs which are known to govern this tuberization phenotype. This pathway is very complex. We thought it was very easy 15 years back. Now 15 years we have been working on now we see that this pathway is far more than what we thought before. It's very complex. There are many factors which are governing this mechanism. It looks very simple, but it is indeed a very complex mechanism. Okay, and many things we have worked. We have been working on this to understand what is this, that how they crosstalk, how they signal, how are the major hormones play in this tuberization phenotype. I will move on and will show you this. So please see, look this. This is the question that we asked long back. What is that signal? Why would this plant, it is now in the mature stage. It is now have lot of mature tubers. And what it happens, please look at this figure. If it senses light, this hook-like stolon, it makes another plant. So for me, this hook-like stolon is nothing but a shoot-apical meristem. Just try to understand one fact. This stolon, if it doesn't sense light, it grows a tuber, and that's what the conversion is. This stolon to tuber transition is a dynamic phenomena, is a dynamic process. That's what we believe today. Because we have identified huge number of regulators that can control this process in the early stages of stolon development. And what I'm trying to tell you today, that this is governed by an activator repressor module. Because we have an activator in our hand, we have a repressor in our left hand. And we see how this activator repressor module control this phenomena in the tuber development process. Okay, so let's proceed and see. Tuberizations is a reproductive phenomena like flowering, and as I said, florigen and tuber. This is no more florigen, by the way. This is now known. It's a FT, flowering time MR protein, which is now a mobile signal for the flowering. We now know tuberigen, what it is. We have ST-BEL5 and ST-SP6. Two major signals have been discovered in order to understand that what is governing these plants to develop the tuber phenotype. Okay. To cut long story short, I will just try to say one more thing is that this plant makes, the plant that we work on, it's a photosensitive. It senses light. It does not make any potatoes as soon as you have a long day duration light. It will only make potatoes when you have a short day light, last duration light. Please remember this point. It will help us to understand. And this is if you put them in long day, we do not get any tuber. So that makes the model organism very fantastic to understand the light and its role in the developmental process. I said again, this is the review from the pathway. These are all these regulators we have now almost worked out and tried to understand their role. I will just talk about today the 3 mobile RNA that is governing this process, and I will try to come back to this quickly. This is the one that we identified long back, BEL5 protein. Okay, it's known as potato protein, Solanum tuberosum. It's called BEL5 protein, ST-BEL5 I say, and BEL5 interacts with POTH1. These are NOX. NOX are master regulators. Entire plant architecture is governed by NOX, like HOX in Drosophila and NOX in plants. Okay, so these two interact. And we knew this before, long before 2003, we knew that these two interact. So are the other BELs and NOX. And this BEL and NOX interaction is very critical for all types of plant architecture and governing the complete development process in the plant. Okay, so what we did, I will show you few slides from pretty old of my work. We did this BEL5 RNA accumulations in the plant under 2 different light conditions, short day versus long day, and we see huge amount of RNA accumulated in the short day. If you look at the RNA accumulations of BEL5 in this plant, and you see compared to long days in short day, lot of RNA is accumulated. So you ask a question, why do you see such a RNA accumulations under a short day photo period? Okay, maybe there is something going on in these plants. We overexpress this BEL5 in potato, and what we see that plant makes more number of tubers. That's the message. We overexpress BEL5 in potato. We see plant makes twice the number of tubers. Tubers shapes, size we are not worried about. We see more numbers and more in terms of fresh weight as well. Okay, plants looks pretty normal. If you compare with the wild type, they look very normal. So there was no concern from our side. We knew that if you overexpress, it makes more tubers. That's it. Now, if you reduce, if you make an anti-sense line, if you knock it down from the plant, what you like to see? If you knock it down, you look at what happens. You have roughly about 30 to 40-fold reduction in the tuber yield. If you overexpress, it makes more tuber. If you reduce it, it makes almost no tuber, you can say. So is that if it's all the lines we checked, we look at the target tuber fresh weight, and we see the tuber yield is reduced in this line. We did a grafting experiment long back, and what we like to show here, this is my overexpression line, and if you graft this line onto a non-inductive potato plant, and if you see, just quickly look at the graph that I did, and this plant, by the way, did not make any tubers. When you grafted this plant made a lot of tubers here. This is after almost 2 months of observations. Almost 2 months of observations we see. That means something has moved. And that was the puzzle long back. It is no more a puzzle now. This technique is now pretty standard in the plant biology. People do this very routinely now to understand is there any mobile signal. And we did not know what was it. But there were old reports in 1980s and 1985s. We finally really nailed down that what it is. We came down the conclusion. It is the BEST-BEL5 RNA which is moving from here and triggering the plant to produce tubers. So BEL5 is a positive signal for tuberation. It's an activator. It activates the process. Okay, fine. That's the BEL5 all about, old story, right? All these are already published. Nothing new here. You can look at all this. We know where does BEL5 express. To understand this BEL5 expressions, we did a lot of promoter activities, and to identify that where does it express only in the veins, in the phloem, in the companion cells, as well as in the phloem parenchyma, making it more strong signal that if it is a mobile signal, it must be expressed in those tissue types for it to go from leaf to the stolon. That was the time when people showed that. So now I told you that this condition is inductive. If you put the plants in this condition, plant makes a lot of tubers. It's a non-inductive, right? What happens in these two conditions? In non-inductive conditions, you have another protein known as REP1. REP1 level goes high, and REP1 binds to STBEL5 promoter. It is repressing STBEL5 transcription. As a result, whatever STBEL5 is made here is not sufficient to trigger the tuberation. On the other hand, you have the short day. You have ahead of REP1, you have a microRNA 172. MicroRNA 172 cleaves REP1. As a result, BEL5 is free. BEL5 can easily transcribe in this condition, and it can move from here after transcriptions to the target organ and induces the battery of the tuberation circuit. Okay, that is clear now. Okay. I just want to show you just one slide that we have also shown recently, that it is not the 72 alone. There is another microRNA upstream of 72 known as 56. We worked extensively on 56, and we showed that microRNA 56 induces aerial tubers, although the tuber numbers are less, but it produces a lot of all the stems which are likely to grow below ground. All these stolons, they grow from the axils, and plant have a beautiful phenotype. Okay, although the tubers are small in size, but plant have a beautiful phenotype. We are still investigating this mechanism. I'll move on, and I'll try to come back to the main story, that how does this RNA move? Now, this RNA is transcribed here. It can go this way, it can go this way. There was absolutely no clear picture in the field. 2009, the first paper came from UC Davis. They showed that in cucurbits, the pumpkin, there are as many as 16 RNA protein cans binds to an RNA molecule, and it can take it to the up. It has nothing to do with the gravity. Please do not confuse yourself. This is completely a selective guidance mechanism plants have to execute based on its need. Okay, so after this paper, we looked at the potato and we identified those proteins, the orthologs in potato we identified, and then we did lot more work. Okay, so this was the case that these proteins bind to this RNA molecule, and this proteins has polypeptide RNA binding, RNA recognition motifs, RRMs, and they can bind to it, and this complex known as RNPs, ribonuclear protein complex, and this complex can go from cell to cell. Imagine it's a long distance transport in case of potato, because it is I said BEL5 expressed heavily in the leaf. After expression in the leaf transcription, there is not much protein is made, very less. We have seen carefully in the leaf that protein is less, and this RNA then moves to the stolon, the below ground organ. What is that? After that, in between, I think I'm missing, yeah. So I did one more experiment and I looked at the Bel5 RNA very carefully, and I see that Bel5 RNA has its, any RNA will have its 5' and 3' UTR. If you look at the Bel5 RNA, it has loaded polypeptidin tracts in both its 5' and 3' UTR. I said, fine, maybe that is making the case for me, for in our case, because we do not know which is the protein